Hello, everybody. Happy Tuesday. Thank you for joining us again for another live virtual classroom. This week, we are talking about a wide range of things, but today specifically, we're talking about the scientific process and how it can be used to find out how much toxins are in our sharks. Usually, we have a education ambassador do these shows, but we're doing a little change up today. Today we have Lisa Crawford, who's from Stony Brook University, and she is one of OSearch's collaborating scientists, and she's going to be leading things today instead of education ambassador, which I think is going to be really exciting. I think you're all going to like her. So hi, Lisa. Thanks for being here.
Hi, John. Thank you so much for having me. I'm really excited to be here with everyone.
Cool. So just so everybody knows, there is a comment section in this, or just below this video. If you guys have a question, let us know. The more on topic your question is, the better chance we'll have of answering it. But yeah, we hope you take advantage of it. So since Lisa, you're kind of in the driver's seat, I think I'll turn it over to you. But first of all, I think you know who I know you know who this shark is. Who is this?
Yeah, this is white shark Luna, who we caught in Nova Scotia in 2018, showing us her toothy grin.
Yeah, what a night that was. You remember that night?
Wonderful night.
Gosh, she was pretty. She was one heck of a feisty shark. But
that's a feisty girl right there.
Yeah, that's one we'll remember. So just so everybody knows, Lisa is out on the ship with us. She's down on the platform, mainly taking muscle samples, but she'll also be down on the platform helping out with all of the other researchers. I guess I say that just to say, she knows her stuff. She's there. She's on the ground with us all. The other cool thing about Lisa is, expeditions can be long. Day 17, 18, things start to become a little bit of a grind, but there's one person who kind of picks things up and helps put a smile on everybody's face, and that's Lisa. So not only thank you for being here, Lisa, but also thank you for the energy that you bring to OSearch as a whole. Anyway, so with that, I will let you talk about the scientific process and how you use it on a daily basis.
Great. Thank you so much, John, for the wonderful introduction. I am so happy to be with all of you today. I love talking to kids. You guys come up with great questions, so please think of some good ones and put them down below in the chat, and we'll get to them at the end. So to kick things off, I just wanted to let you guys know that I am a student just like you. I still go to school, I take classes, I study, but I'm also a scientist. And as it turns out, being a student and doing science are my job. And I study aquatic toxicology. So you would call me an aquatic toxicologist. And there's a picture of me doing some of my research on some sandbar sharks. But we're here to talk about white sharks today. So let's.
Is that an aquarium?
That is actually at Moat Marine Laboratory down in Sarasota, Florida. In a huge tank they have that's not part of their main aquarium, but it's part of their shark research area.
Yeah. Cool.
Yeah. So to kick things off, I do research for my job. And just to make sure we're on the same page, what is research? Well, research is simply gathering information or evidence to help us answer questions. We can do research in everyday life. If we find things around us that pique our interest, that make questions develop in our minds, we can gather information to answer those questions, and that's research. I happen to do scientific research. And well, you're probably wondering, how do we do research? Well, there's actually a very simple method to doing research that I'm going to walk you through, and it's called the scientific method. So it starts out by making an observation. So these are things that we see, hear, taste, smell, and touch, things we're observing with our senses that make us curious about the world around us. And that leads us to ask a question. Why is something happening? When is it happening? What's going on? And then we can form a prediction based on what we already know. So I think this is happening because, or I think this is happening at a certain time because of this other information I know. And then we can gather some information, gather some evidence, collect data about our question and our prediction to help us finally answer that question once and for all. And so this is the very simple process of the scientific method and doing scientific research.
So before you go on, I got a quick question though, Lisa. This is something that you use on this is how you answer your big complex scientific questions like poisons, well, toxin. We'll get to the vocabulary here in a sec. But people may do this process a little bit more on a daily basis than they think. The question and answer. So walk me through an example. Like, let's say I open up my fridge and I'm like, well, something's gone bad in here. How can I this process can help me figure out, there's something funky going on.
Yeah.
And then get to the root of the problem. So can you walk me through how this process can apply?
Absolutely. So yeah, just like John was saying, you can use the scientific method at home with things like something you smell, like a stinky refrigerator. So that's your observation. You smell something stinky in your fridge. And so that leads you to a question is, what smells stinky and why does it smell? And then you can make a prediction. It's like, well, that lasagna that we made has been in the fridge for a couple of weeks now. And that's information I already have. And so now we're going to do an investigation. We're going to gather some information and some evidence. We're going to go into the fridge. We're going to look at all the food in there and figure out what is causing the smell. And sure enough, we can find the culprit. It's that stinky lasagna. And so that answers our question and confirms our prediction that we made. And so that's a really fun, easy example of how you can do scientific research at home. So yeah, this process is so simple that we can extrapolate it onto bigger things like I do for my scientific research on sharks.
But your hypothesis isn't always necessarily correct, is it? Say it's like, I think it might be that lasagna. And then you go and you get to the lasagna, you're like, no, that smells fine. And it turns out it was like a bag of salad or something. Yeah. Or so in your research and more real world, you think the toxins or poisons are coming from somewhere else. So the hypothesis isn't always correct. Am I right?
That's absolutely right. So part of our prediction making, we generate what's called a hypothesis, where we can ask our question and confirm or deny whether our prediction was correct based on the information we had. And sometimes your prediction is right. Your hypothesis is confirmed, and sometimes it's not. So yeah, if you went and did your investigation of the funky refrigerator and you looked at the lasagna and the lasagna was perfectly fine, you would have to say, nope, our hypothesis was not correct. Our prediction was not correct. It was in fact the old bag of salad. But hey, that's new information that we have. And we answered our question.
Sorry. So let's move out of the fridge and into sharks. And how does that sound?
Into sharks, yeah. So going back to what I do, like I said, I'm an aquatic toxicologist. So I research toxins in the water. And toxins are just poisons. They're poison chemicals that make their way into the water. And then more specifically, I research the impacts of these toxins on sharks. So once the poisons make their way into the water and then eventually make their way into sharks' bodies, I look at the impacts of those toxins on sharks. And so a vocab word I want to give you today is toxin. A toxin is a harmful chemical or a poison. And so now you're wondering, what does that have to do with white sharks? Well, white sharks can get toxins in their bodies. And so these toxins are tiny little molecules that go into the water and then eventually make their way to the ocean, where they're eaten by plankton, that are eaten by small fish, eaten by bigger fish, and then eventually white sharks. And so if all of those animals have had toxins in their bodies, when the white shark goes to eat that last fish, it gets the toxins in its body too. So you can see here from this simple diagram, we have a fish with toxins in its body that's eaten by the shark, and then the shark gets the toxins in its tissues, in its blood. And unfortunately, toxins are really hard to digest and metabolize and break down, so they can't easily be excreted. So over time, as the animal grows and ages, it accumulates or gains toxins in its system. And so another vocab word I want to give you is bioaccumulation. And so this whole process is called bioaccumulation, where living organisms accumulate toxins in their bodies from the food they eat. And this is super important to understand when it comes to white sharks, because white sharks are apex predators. They're feeding at the very top of the food web, so they're potentially eating a lot of contaminated prey like seals and tunas. And it's also important to remember that white sharks can live for a very long time. They eat a lot of food and they can grow really big. So they have the potential to bioaccumulate a lot of toxins in their systems.
So real quick, Lisa, maybe you'll get to it later, but a couple of people are asking the question right now. How many toxins, Megan asked, how many toxins do you find in the average shark? Is that even something we can answer right now? And Bela has a similar question.
Yeah, so that's an interesting point to make, is how many toxins are in the body. What I have to do is identify toxins that I want to look for. So when I'm doing my experiments in the lab on the samples I collect, I have to know, oh, I'm looking for mercury, I'm looking for PCBs, I'm looking for pesticides. So unfortunately, the methods that we have to do toxicology won't reveal everything to us. We have to put our investigator cap on and go in and look, make a prediction, make a hypothesis about what we think is in the shark's body, and then use the evidence we gather to confirm, oh yes, those contaminants that we thought would be there are in fact there.
So Megan and Bela, the answer is we don't necessarily know exactly how many there are. We have to think of toxins that might be there first, go and look, see if it's there. If it is, we can put a check mark. But obviously that means a little bit of, maybe there's something that we're not thinking of to look for and it's there. So that's why we can't answer the question. So yeah. Go ahead, Lisa. Sorry, I just wanted to make sure I got to those questions real quick.
Yeah, no, that's a great point. But luckily, to answer your question further, there are lots of other toxicologists in the world who have done similar studies with other types of fish. And so they give us a pretty good idea of what we should be looking for in the white sharks. So now, to get more specific on what exactly is my research, well, let's go back to our scientific method flowchart. So the observation that I've made and that we've talked about already is that white sharks are apex predators feeding at the top of the food chain that eat toxic prey. So that's going to lead me to a big question, which are, what are the levels of toxins in a white shark's body? And potentially, like y'all were asking, which toxins are in the white shark's body? And I'm going to make a prediction that because white sharks are long-lived, slow-growing apex predators, they're going to have very high levels of toxins. And we can use what we know from other scientists' work to give us a list of contaminants to look for in the shark's body. So then, in order to help us answer our question and confirm our prediction, our hypothesis, we have to collect tissue samples, gather information, and record data. And finally, that will eventually help me answer my question. But I'm still in the process of doing all this, and that's where OSearch and the white sharks come into play. So my fellow scientists and I go out with OSearch. The fishing team catches white sharks that we can then study and ask a lot of questions and try to answer them with the scientific method. But one thing that we all have to keep in mind when we're working with white sharks is what kind of data, what kind of information is important to collect, and which variables do we need to measure. And this brings us to our next vocab word. What is a variable? A variable is a type of data that can change, a type of information that you might gather that would be different from animal to animal or place to place. And rack your brain. Can you think of any examples of variables that we might want to measure when we're working with white sharks? Think about something that would change from white shark to white shark. I'll give you a few to jog your mind here. We've got things like gender. Is the shark a boy or a girl? Not everyone that we catch is the same. The latitude, where in the world did we catch the shark? What are its stress levels? Those change from shark to shark. Some sharks are more stressed than others. We've got other variables like body circumference. That's something important that I have to measure because remember I said that animals, as they grow bigger, they gain more toxins in their body. So body circumference is a very important variable that I have to consider in my toxicology work. And then, what would you know? Toxin levels is also a variable, right? Think about it. From shark to shark, the toxin levels are not going to be exactly the same. They're going to change. So that's a variable that we can measure. There's more. Age, food preference, diet, water temperature. These are all variables that are going to change. So I've put a bunch here up on the screen, but I'm sure you guys are smart. You can probably think of a whole bunch more that I didn't think of. So I'm not just working alone with OSearch. I'm working with a whole team of scientists that's trying to answer a lot of research questions. And so when I'm on the lift, I'm focused on my work, but the entire team of people is also gathering information, collecting data, and measuring variables to help them with their scientific research. Now, I wanted to walk you around this picture here to give you.
I was going to say, there's a lot going on in this picture, Lisa. You're going to have to help break this down a little bit for us, since it looks like everybody's kind of got a job to do.
Yep, yeah. Everyone in this picture is doing a very important job. When we have sharks on the lift, there's not a moment to lose. Every minute is critical, and we have to gather as much information and measure as many variables as we can. So let's start with Captain Brett, who's on the far right side of the picture. Captain Brett, when the shark is on the lift, stays at the head of the shark. And he's looking for things like the water flow of the hose into the shark's mouth. Is the shark breathing? Is its body condition okay? So that's information that he's gathering and variables that he's measuring. Then we'll move on to veterinarian Dr. Mike Hyatt, who is in charge of doing an ultrasound. When Mike is doing an ultrasound, he is gathering information on whether the shark is pregnant, are the organs healthy, and then a variable that he can measure is the white shark's heart rate, which is pretty cool. And then standing right behind him in the picture is my good friend, Dr. Lisa Hoops. Dr. Hoops collects blood sometimes when she's on the lift. Blood is super important because it can help us answer a lot of research questions, and there's a lot of variables that we can consider. Blood can tell us about the stress levels of the animal, its nutrition, what it's been feeding on, its genetics, so who it's related to, and it can also help me with my toxicology work. All right, next we'll go around the circle to DJ, the OSearch first mate, who is standing there at the very back of the shark. DJ helps when the shark is on the lift to hold the animal's tail, make sure the animal is under control, and keep all the scientists safe. What DJ is looking for and the information he's gathering is things like the shark's muscle contractions. Does it feel healthy and safe? And he's also looking for the body color of the animal to make sure it looks healthy. So color is another variable that we can consider. And then we'll move on finally to Fernanda in the blue hat. Fernanda helps us collect the data, record it, and she writes down all the variables that we are observing and measuring while we have the shark on the lift. Very, very important variables like, is it a male or a female shark? Is the shark doing okay? Where did we catch it? What's the weather? What's the sea surface temperature? So she is recording all of that so that the scientists can then use it later to help us with our scientific questions.
Quick thing here, Lisa, going back to the picture. Yes, that is. Someone was asking if that's DJ in the blue hat, blonde hair. Yes, that's DJ. And then one person asked, "Are we collecting urine from the shark?" Mm-hmm. And that's opportunistic.
Yeah. Yep. So urine is not something that we usually catch from or collect from the sharks on the lift. They don't have a typical urine stream like a human would, so it's not something we can easily collect. But there are other fluids from the shark's body that we do collect that can tell us about its urogenital system and how its internal health is doing.
I have seen one that did start to urinate on the lift, and if that happens, yes, we take the sample, but it's not one that we normally get. And then there's another question here. All of this takes about 15 minutes. The shark is not about the cutoff is 15 minutes. Any of these processes that aren't done in 15 minutes, the platform goes back down into the water and we let the shark go. So that is our hard cutoff for how long this process takes. So it's a very choreographed thing. All of the researchers, like Lisa, they all talk ahead of time. They know exactly who's doing what so that there's no missed time while we're on the lift.
Absolutely. Yeah. Good questions. Okay, so now I want to talk about some of the variables that we can measure when we have the shark on the lift. And one of the most important ones, at least for my research, is body measurements. So actually measuring the length of the shark, the circumference of the shark, the distance from the tip of the nose to the tip of the tail. Because remember, going back to that vocabulary word I taught you, bioaccumulation, as these white sharks grow bigger, they accumulate more toxins. So it's important that I know exactly how big the shark is. And you can see in this picture here, we have a tape measure stretched over the back of the shark that Captain Brett holds one end of the tape measure at the shark's snout, and we stretch the tape way out to its tail, and we take several measurements of its body length, and then we wrap this tape measure around the middle to get its body circumference. So that's a variable that we measure for each shark that comes on the lift is body size, body circumference. And this picture also is great to show the spot tag that we put on the fin. So that's that black rectangular thing you see on the shark's dorsal fin there. We're going to talk about that right now. So the spot tag is probably the most famous piece of OSearch technology that you've all seen because it helps us get the information that goes up on the OSearch tracker. So the spot tag goes on the shark's dorsal fin like you saw, and every time the shark comes to the surface of the water and sticks its fin out, the spot tag can communicate with a satellite and tell us exactly where in the world the shark is. And so when you go to the OSearch tracker, you can see exactly where the sharks have been. So in this image of the east coast of the United States and the North Atlantic Ocean, every single yellow dot is a place where a white shark has come and stuck its fin out of the water so that the spot tag could communicate with the satellite. And then we can connect the dots for each shark to see where they've traveled and make up a new hypothesis about where they might be going next. So I've talked a little bit about samples. And as you saw in that picture of all the scientists on the lift with the shark, everyone was taking samples. So I want to talk about what a sample is and what types of samples that we take. So as this image shows, we take over 12 different types of samples in 15 minutes. Yeah, that only 15 minutes while the shark is on the lift. And we take samples from all over the body. So one of the critical samples is blood. Blood can give us information about if the sharks are reproducing, if they're stressed, if they have a good level of health, if they are eating properly, if they have good nutrition. And then I can also use it to help me find those contaminants and measure the levels of toxins in the sharks' bodies. So this is a wonderful picture of my dear friend and OSearch veterinarian, Dr. Harley Newton, collecting blood from the tail of the shark. And you're probably wondering, why do we take the blood from the underside of the tail of the shark? Well, if you were to go to the doctor to get a health checkup and the doctor would need to draw your blood, they would probably take it from your arm. Well, sharks don't have arms, and as it turns out, their fins are not good places to collect blood from. And in fact, underneath the tail, there are some pretty big veins that give us good access to collect their blood. So you can see Dr. Newton here laying down on the lift, getting beneath the tail so she can collect those critical blood samples that go to help a lot of OSearch researchers answer their questions.
Do you use the blood? Because I know that mainly you take muscle samples.
Yes.
But I mean, from what I understand, there's a lot you can learn from the blood.
Mm-hmm.
So is it something that helps you to decide whether there's toxins or poisons in a shark?
Yeah, that's a really good question, and it actually digs in a little bit deeper into the toxicology. So certain toxins end up in different places of the body. So I mainly focus on muscle tissue because a lot of the toxins that I'm looking at end up in the muscle, but there are toxins that bioaccumulate very heavily in the blood. And so I'm collecting both muscle and blood so I can see that full suite of toxins. And like you were getting at, John, because so many other OSearch researchers look at blood for their work, those are variables that I can then bring into my research. Variables like stress levels, nutrition, genetics, the relatedness of the sharks, diet, what they've been eating. And those are all things that can help me answer my questions. So blood is liquid gold for us. Absolutely. And then finally, that's a perfect segue into the sample I collect, which is a muscle biopsy. So I take a teeny tiny pea-sized piece of muscle from the shark's back. So small, they don't feel it, they don't notice it. But that piece of muscle is critical because that's really where I'm gathering the most information and the most data to help me answer my question, which is what are the levels of toxins in the shark's body? But the good thing is I can share that small muscle sample with several other researchers who are answering questions about diet, genetics, nutrition, and all sorts of things like that. So every sample we get is precious, not just because we have to collect it quickly and we want to make the most of every shark we get, but they're also important because we're sharing them with so many scientists who are trying to answer a lot of different questions. And here is just a picture of me when I'm on the lift, taking my muscle samples. It's one of the things that I'm usually in charge of when I'm on the lift with a shark. That's my priority, get my muscle samples for my research, get them in collection tubes safely and securely. And then once the shark leaves the lift safely and we go back into the lab, that's when I divvy out pieces of muscle for the other researchers so they can do their work too. And so that kind of wraps up what I wanted to tell you guys today. I want to thank you all for listening. I hope you learned something. And I'm really, really, really excited to hear your questions.
Yeah, there's some good ones here that I wanted to jump to. Kira Dolly, I think I'm saying that right. It's not really a question, but she says that back when you were talking about the scientific process and the steps that you take, it sounds like she and her mom were using the tracker to kind of do a similar thing. They would look at sharks and then make an estimation about where that shark might go next. That's using the tracker to basically do the exact same process you just talked about, isn't it?
Yeah, that's awesome. That's really great. So you're using the information that you already have from the tracker to know where the shark has been to help you predict where the shark is going. And that's a hypothesis you can make. So based on the information that we have, which is where the shark has been, we can then hypothesize where it's going to go. And then eventually we'll hear from the shark, it will ping the satellite, and we can confirm our hypothesis. So yeah, that's awesome that y'all are doing that.
Yeah, I mean that's exactly why we have the trackers, so that here's the thing, the tracker is live. Everybody is seeing it at the exact same time that we do. If you happen to jump on and you see that your favorite shark, whether it's Miss Costa or Luna or whoever it might be, is somewhere and you log on before we do, then you know where that shark is before anybody at OSearch even does. And we keep it that way so that we can encourage exactly what Kara is doing in terms of solving this project with us. So everybody out there, go ahead and take a stab. Maybe your hypothesis will be right and maybe your hypothesis will be wrong. But I mean, that's part of the excitement of it, isn't it, Lisa? Like,
absolutely. Yeah. The excitement is the unknown, but being able to be a part of the scientific process and come up with a hypothesis for what's going to happen next. And I applaud you for taking the initiative to do some OSearch scientific research at home. There's other things you can do with the tracker too, like calculate the speed that the shark is traveling. if you have timestamps from 2 points where the shark has pinged the satellite, you can calculate the speed it's traveling, and then maybe that can help you generate some hypotheses about how fast sharks travel. Do all sharks travel the same speed? So there's a question for you guys to answer at home.
Question for you. This wasn't one for many of our viewers, but we just talked about hypothesis and going through the process, and sometimes you're right and sometimes you're wrong. I mean, as a researcher, you've done lots of experiments. Is it upsetting when your hypothesis is wrong, or?
It can be. You know, we spend a lot of time developing those questions and those predictions, and you know, we make a hypothesis because we think it's going to be correct. So sometimes when you get to the very end and it's not correct and you kind of have to maybe start over, it can be a little discouraging, but that's also part of the excitement. Is it's an ongoing process. Now, for me, in my work specifically, if I'm looking for a specific toxin or I'm expecting to see a certain level of toxins and then I don't see those things, well, that's actually a good thing. You know, we don't want these sharks to have high levels of toxins in their body. So sometimes for me, if my hypothesis is not correct, it's actually a very good thing. So that can be encouraging to me, and encouraging that the oceans are doing okay.
So that's a good segue to a next question, and you put up the vocabulary word toxin, which basically means poison. A couple of people have asked, as you've been doing this, and your research isn't quite done yet, but is there one particular poison that you are finding in the sharks more than any other ones?
Yeah, so to kind of get at that question, the toxin that we're looking for the most and we see in the highest levels is mercury. And
somebody did ask what mercury was. So
yeah. So mercury is an element. It's just a naturally occurring element. And it's released through natural processes like forest fires or volcanoes, but it's also released by humans in the form of coal burning. And the more coal we burn, the more mercury ends up in the environment, and then eventually that mercury ends up in the ocean, where it can become a form called methylmercury. And that's the molecule, that toxin, that can actually end up in the shark's body and start to bioaccumulate and cause all sorts of issues to its health. Yeah. But in addition to mercury, we're also looking for other toxins like pesticides. So pesticides can run off from your yard or from farmland and end up in the ocean. They also bioaccumulate. And then a new toxin that we're going to start looking for, but we haven't done yet, are what we call PFAS. And it's just those letters, PFAS. And PFAS refers to things like nonstick coatings. So any type of packaging or surface that we would call non-stick has these chemicals called PFAS molecules on them that when you rinse a pan or rinse a bottle that has it, it ends up in the water, eventually ends up in the ocean. And so that's kind of an emerging toxin that we want to keep an eye on. But in the coming expeditions, hopefully later this year, I will be looking for PFAS in the white sharks.
So Luke asks, we're looking for these toxins and we are finding that some of the toxins are there. Is it making the sharks sick? I mean, obviously there's a big population of sharks. They're all, seem to be doing okay. Are these toxins making the sharks sick?
Yeah, we don't know. And that's the $1 million question of my research right there, is what are the health consequences? What are the effects of toxin bioaccumulation on sharks? We simply don't know yet. And so a lot of my work is very exploratory. One of the things that I didn't mention for the scope of this presentation is that the muscle samples I collect, I also use them to measure gene expression. And so what I'm looking for there is to see how the cells of the shark are actually responding to toxin exposure. So that's going to help us start to chip away at that question, is what are the actual health effects of toxin bioaccumulation? But we have to start at the smallest biological level of organization, which is the molecular and cellular levels, to help us start to build that chain of understanding as to how toxins are affecting the entire white shark and how it's affecting the white shark population. And it's really great that I have the opportunity to work with OSearch, because I'm not alone in answering this question. All of the researchers like Dr. Newton, Dr. Hoops, who are studying white shark health can help me, and we can help each other answer this question simultaneously. So some of the variables that they are measuring, like stress levels, nutrition, diet, those variables help me start to answer that question directly of what are the impacts of chemical contaminants on white sharks. And in a couple weeks, we'll actually be doing a live stream like this all together, talking about our work. So stay tuned for that.
So as we start running out of time here, we'll make this kind of the last question, because it's a good one, and it kind of, I think, helps wrap things up a little bit. I forget who asked it, but it was asked a little earlier on. Can you talk about, so if there's toxins in sharks, could that have an impact on humans? Could those same toxins make humans potentially sick too? And if so, how?
Yeah, so there has been a lot of research into this, and that's a very good question and a good point to be aware of. We eat a lot of the same things that sharks are eating. We eat fish that they eat, and so if they're eating contaminated prey, so are we. And so that's why when you hear about things like limiting your seafood consumption because of mercury, that's why. Because it's not just the apex predators, the white sharks, who have toxins in their body. It's all the other fish in the food chain too. So if we're eating the same diet as the white shark, we also have the potential to bioaccumulate those toxins. So don't eat too many sushi dinners. But the good thing there is that because we do tend to study humans a lot more and we have a good understanding of human health, we actually know what happens to the human body when we ingest too many toxins. And so there is a little bit of safety there. You don't have to be afraid to eat seafood and have toxins in your body too much, as long as you're limiting your consumption of high-risk seafood, so things like tuna, tilefish, and choosing mercury-safe or contaminant-safe fish. And there's plenty of resources online that can help you choose seafood that would be low in toxins that's perfectly safe to eat.
Cool. Yeah, I did not know that about mercury. Well, we hope that everybody enjoyed this. We hope you learned a thing or two about the scientific process, whether you're using it for something as big and complicated as looking for poisons in sharks like Lisa is doing, or you're just investigating things around your own house. Using the scientific method is an extremely important thing that we do on a regular basis, whether we realize it or not, to just solve problems. So Lisa, thank you very much. Do you have anything else you want to say before we sign off here?
I just want to thank everyone for tuning in, listening. I loved your questions. They're very thought-provoking. And so thank you for being here, and keep following the OSearch Sharks and keep doing research in your own home.
Yeah, thanks everybody. We plan on doing a few more of these virtual classrooms, so keep an eye out on all of our social media channels for details on when and where those will be. And please keep up the great questions. There were a lot of really good ones today. Love seeing so much great engagement. That's why we do these. So to everybody, thank you. And with that, we'll sign off.
Thank you.